Our presentation today will be delivered by Dr. Michael Kobor on the topic of epigenetics and the human life course. Dr. Kobor has been at the University of British Columbia's Department of Medical Genetics since 2005, where he has built an interdisciplinary team focused on developing a society-to-cell paradigm for population health research. Dr. Kobor is a renowned biomedical researcher who studies how our social environments and life experiences get under the skin through biologic embedding to influence health and behavior across the lifespan. As one of the world's foremost experts in social epigenetics, he leads a program of interdisciplinary research with the goal of translating foundational genetic discoveries into interventions and policy change to promote healthy life trajectories. Dr. Kobor is the inaugural Edwin S.H. Leong UBC Chair in Healthy Aging, a UBC President's Excellence Chair, where he is leading a new center focused on healthy aging, uniting faculty, trainees, and research personnel across UBC and engaging experts at universities around the world, where he will lead new collaborations on a wide range of interrelated projects. The new Edwin S.H. Leong Centre for Healthy Aging also aims to help us better understand the combination of genetics, environments, and lifestyle choices, and how they help determine health and aging, and how we might effectively intervene at any stage of disease vulnerability or disease progression to redefine the aging experience and improve quality of life. I ask everyone to please hold your questions to the end, where we'll be moderating a short discussion period before we end at about 1 o'clock. Thank you, Dr. Kobor.
Well, thank you so much, Al, for the very kind introduction. And it's wonderful to be back in Toronto. I spent 7 years here doing my PhD. Went to a baseball game yesterday. It was very exciting for those of you who follow the Blue Jays. And so, welcome everyone. Dr. Leong, great to see you again. It's been a while, 3 days, I think, isn't it? Closer? Okay. So what I'd like to do today is, first of all, acknowledge that the work at UBC is really done, it's situated in the traditional ancestral and unceded territory of the Musqueam people. And it's a privilege and a joy to work, play, and learn there. So today I was planning to talk to you, as I said, about the biological vestiges of early life environments and experiences, how they get under the skin. I was planning to tell you a story about epigenetics and Parkinson's disease of mice and men. We might skip that because Ayal tells me I have to be finished at 12:50, and so and then really talk about the epigenetic age, which is something that we're really excited about these days, and leading from there into an invitation to all of you to learn more about the Edwin S.H. Leong Centre for Healthy Aging that we just launched at UBC last week. So with that said, what is epigenetics? Well, as you probably already heard, I grew up in Germany, and Germans love definitions. I actually hate definitions, which is why I'm in Canada. So for those of you who like definitions, this is probably one of the more complete definitions of epigenetics. What we're really interested in is the social part thereof, and that is the idea that as we go through life, there are experiences and environments that influence the way our genes are expressed. And we're thinking this in a fairly simplistic way. There are these experiences. They get under the skin to affect biological pathways of various sorts. And we're going to talk about a few of those today. And then ultimately, in some cases, drill all the way down into the nucleus of the cell, where they might affect the level of gene expression, potentially in a lasting manner. This, of course, is not a one-way road. You can easily then imagine this going the other way as well. And so we're going to focus today on the left part of this paradigm. And for those of you who are not experts in gene expression, it's really at the heart of epigenetics is the idea that, contrary to this little drawing, the DNA in our cells does not occur in its naked form, but instead it's packaged into multiple ways. And my former postdoc, Maria Aristizabal, who is now at Queen's University, she recently published a very comprehensive review in PNAS that outlines the complex landscape of these, what we call epigenetic marks and mechanisms that are really, as I said, rooted in the fact that the DNA is packaged in many ways, You can see here some of the canonical modifications like these histone proteins that pack the DNA. There's also, of course, RNA modifications and non-coding RNAs that play a role in all of this. In our lab, especially on the sort of human life course side of things, we almost exclusively focus on DNA modifications. And these come in various forms and flavors. Most often, there are little small chemical additions called methylations to cytosines in the DNA. And again, even those come in 2 different or 3 or 4 different forms. There's also methylation at other nucleotides as well. And so collectively, these occur in all of our cells and shape that landscape that is basically the template for all of this epigenetic biology. And again, we look at this very much in a life course perspective, because these things actually change as we go through life. And to make it very simple, I always love to think about all of this complex biology and all of these complex relationships just by thinking of a light bulb and a dimmer. And so the light bulb in this case would be actually the green one, the level of mRNA, so the gene expression, if you wish. And the dimmer is really this epigenetic marks. And so how can we think of this in the context of a single gene? I know, Steve, what's your favorite gene?
Favorite?
Yeah.
Cystic fibrosis.
Cystic fibrosis. So you have your favorite gene, and so this is the DNA, again, in its naked form in this case. And what happens is that we can have this gene either totally on, so the light bulb is totally on, or totally off. Or if you live in a fancy house, you have a dimmer and you can set that light bulb in different ways. And so what the epigenetics chromatin, the DNA methylation does, in particular, when it's located in the gene regulatory region, it actually regulates the activity of this light bulb. So how can we think about this? So in this case, the cystic fibrosis gene is totally on. Now you see there's a little methyl group that's being attached chemically to the DNA, pushes that dimmer down. The next one comes on, pushes the dimmer further down. And now we have entirely methylated our cystic fibrosis gene regulatory region to basically end up with a gene that's turned off, a light bulb that's off. Now, it's clear that all of this happens from conception onwards in all of us, in all of our cells. But I think what has really shaped a lot of the dialogue and really the foundation of the social part of epigenetics is that some years, a decade and a half ago, it became clear that the nutrition and the lifestyle of the mother during pregnancy and can affect the epigenome of the child. And the seminal study was done by Bas Heijmans and his group, showing that in elderly folks that were in their 60s that were born during the Dutch famine in the end of World War II, to mothers that had basically almost lived on nutritional deprivation, you can see dimmer changes 60 years afterwards, and as related in contrast to people that lived in the same area but did not starve. And so this has now been replicated many times, many different prenatal exposures, whether it's smoking, nutrition, alcohol, and so on, all associate in various forms and for various lengths with the epigenome of the offspring. And we have done some work that I'm not going to talk about that I presented last time I was here on fetal alcohol spectrum disorders, showing that that is indeed a powerful sculpture of the DNA methylome. Now, perhaps what caused the attention of the public most was seminal work by our colleague from McGill, Michael Meany, who showed 2004 so that the quality of maternal care, initially in a rat licking and grooming paradigm, but later on also in humans, was associated with epigenetic changes in children. And again, I'll show you some of this work that we have continued in some part together with Michael. So that, of course, captured the attention of the public. Many stories to tell about how this could also be misconstrued and create extra stress, but we have to leave that for the discussion, perhaps. And then perhaps more than anything, illustrating the power of epigenetics is if we look at twins. And so you see those 2 twin series, cutie on the right being all happy and content, and then the fellow on the left screaming and shouting, and work from many labs, including Jonathan Mill, have shown that identical twins that basically have more or less the same genome actually acquire discordant epigenomes across the life course, especially if they happen to have been separated early in life. And in turn, that often is associated with various discordance of various diseases, chronic diseases such as heart disease, but also mental health and so forth. Really illustrating that these environments get under the skin to affect health and behavior across the lifespan. So in my own laboratory at UBC, we initially looked at this in the context of epigenetics and child health in that sort of age-old paradigm, nature versus nurture. The nature being our DNA, the nurture being the fertile ground of our families, of our communities. How do they interact to shape the epigenome, and how can we harness the science and knowledge of the critical parameters that shape this relationship to ensure that all our children have a healthy start to life, that we prevent diseases across the lifespan that are rooted in these early life environments. And so over the years, we have had many exciting collaborations. But before we started out of this, we also decided that we want to think about this a little bit in a more nuanced way and really looking at this biological embedding of these marks in a developmental context. And this is a review my former postdoc, Meg Jones, who is now at the University of Manitoba, has written some years ago, where she postulates that while our genome is more or less static, we go through these sensitive periods in life, and many of those tend to be early during pregnancy or the first people's, Sophie, I think you talk about the first 2,000 years, right? And so on, so whatever it might be, but there are all these sensitive periods. And what Meg has postulated, if a particular, say, a harmful exposure happens during that period, it changes the trajectory of our epigenome into this red line that you can see here, and potentially ends up getting under the skin, staying with us for life with a more high risk outcome for particular chronic diseases or health issues. Contrast, when this exposure happens later in life, outside of the spasticity window, it does not affect the trajectory of our epigenome. Now, this is one of the best studied sensitive periods. Sometimes I'm wondering whether it's because we're just looking under the lamppost and there are other sensitive periods. So we're now focusing in part on puberty and adolescence, but also older age. And a lot of this work really has been tremendously influenced and shaped in my own thinking by one of my most cherished mentors, the late Clyde Hertzman, who was at UBC for many years, was a long-term CFAR fellow. And even before all the advent of this epigenetic excitement, has postulated there's a biological of early life experiences that last into effects way into adulthood. I encourage you to read his seminal work if you're interested. So over the years, as I said, we have been collaborating with many different people to really put to test this idea that epigenetics lies at the interface of environments and the genome, or if you wish, of nature and nurture. And we have been focusing in a number of areas, including early life parental stress. So does it matter whether you grow up in a nuclear family with doggy woof woof, or in a family where the parents constantly yell at each other? Does it matter whether you grow up in this nice mansion or in some, not so nice dwelling? So really focusing on early life socioeconomic status. And over the years, as I said, we have been standing on the shoulders of giants, all of whom are listed here. And I can't do justice to highlight all the work we have done with these folks, but I want to just focus on one area, and that is really the lasting effects of early life poverty. And my wonderful research director, Kim Schmidt, who is with us today, published this really comprehensive review recently that talks about how child poverty gets under the skin at the impact of level of child biology, so physiological stress, response to brain structure function, microbiome, as Brett Finley will clearly tell you, immune response and inflammation. We, of course, focus on the DNA methylation. I'll show you some examples of that. But this also then has impacts on child health and developmental outcomes, whether it's cognition, learning, language development, and so on, that you can see in the middle. And more importantly, or most importantly, really has potentially fundamental implications on long-term adult health and well-being, where early child poverty associates with risk for mental health problems in adults and older folks, increased susceptibility to infections, a pro-inflammatory phenotype, an increased risk for chronic diseases, and decreased productivity. So really a powerful paradigm, and if we can change the dial on this, I think we really make a difference and an impact as we go ahead. And so initially with Craig Miller and Edith Chen, some years ago, we put this idea to the test that it indeed is the sort of axis between the dimmer switches and the light bulbs, and initially focus on the light bulbs because the technology to ascertain the dimmer switches as many of you know, wasn't quite up to speed back then, where we had a cohort of healthy adults in the lower mainland in British Columbia that were basically stratified, whether their first 5 years of life were in low socioeconomic status versus high socioeconomic status. And they were mixed in adulthood so that we could not basically look at confounding from adulthood. And what we found in a nutshell is that indeed, when we look at the level of these light bulbs, there are differences in these white blood cells of these folks that are related to whether they grew up in low versus high socioeconomic status. These are not random differences. They were actually predicting, interesting enough, a pro-inflammatory gene expression phenotype. When we then actually put this to test by exposing their white blood cells to agents that engage the innate immune system, so these TLR, things like that you find on top of bacteria and such, what we found is that indeed the folks that grew up in low socioeconomic status had a more higher immune response than the folks growing up in high socioeconomic status. And lastly, at the level of cortisol, so the sort of stress, if you wish, simplistically speaking, again, we find the same, that folks growing up in high socioeconomic status have lower reactivity there. And that is all irrespective of your current socioeconomic status. In work later with Edwin S.H, we then showed that there is a buffering to some extent of this of the resilience by a maternal warmth. So I always tell people, parents can make a difference. Some years later, we then looked at this at a level of the dimmer switches. And again, what we found that the DNA methylation in early life correlated with socioeconomic status. Interestingly enough, in this particular study and in several others since, we do not see a correlation of current socioeconomic status with these dimmer switches in white blood cells. So collectively, really suggesting that at the level of DNA methylation, the light and the dimmers, the light bulbs, the mRNA, the immune system, and the sort of physiological response to stress, there is a embedding of early life poverty. And of course, that portends some really interesting opportunities for intervention. Now, of course, when you do this kind of work, you want to replicate it across different cohorts and across different populations. So more recently, we have been working with Tom Boyce with the University of California and his former trainee, Nikki Bush, who is now the department head of psychiatry and UCSF, in the PAUSE cohort, which is a Berkeley East Bay-based cohort of adolescents, where again, in this case, we actually had a better, a more nuanced measures of various aspects that make up socioeconomic status. So not just income per dependent, but also things like parental education and family adversity. And again, without going into too much detail, for all three of those, we found associations with these 10-year-old children that can be traced back to whether the amount of income, the level of family adversity, and parental education. Interestingly enough, the overlap was perhaps less than we expected, and we're still trying to understand how that is and whether each of those things engages different biological pathways ending up to DNA methylation. So stay tuned with this. Further, we have been going outside of North America, and together with my good friends, Tom McDade and Chris Kusawa, both members of the National Academy in the US, we have been working many papers on the Cebu Longitudinal Health and Nutrition Survey in the Philippines. And again, what we found there, and this is in young adults, that we find particular social and physical environments do associate with DNA methylation, particularly of inflammatory genes. We can also compute an inflammatory index based on these DNA methylation patterns. Unfortunately, they're not connected, as that would have been too nice. But again, showing that in a different population setting with different experiences, different ages, we can replicate this sort of fun conceptual idea that environments get under the skin to associate with changes in this DNA methylation, in these dimmer switches, and potentially for many, many years. So again, I can't do justice to all the various studies that we have done, but there are some of them are listed here. And so we've done this in the context of, again, as I said, sort of family stress. We have done it in the context of actually children who spend the first 2 years in these atrocious orphanages in Romania and places like that, and then were adopted to North America. In our perhaps most extreme study with folks at Harvard, Andrea Roberts and others, we have shown that exposure to childhood sexual abuse is associated with differences in human sperm methylation. And on the good thing and the more hopeful side of stories, we've shown that there's an epigenetic correlate of sort of neonatal contact and attachment and so on. So a really sort of broad body of work looking at very different social environments and their long-term effects. As I mentioned earlier, we've also done some work with fetal alcohol spectrum disorders that I don't have time to talk about today, but with the idea of whether we can harness this biology of DNA methylation of epigenetics to perhaps come up with a better and earlier predictor of fetal alcohol exposure. I'm going to, how are we doing on time? So I'm just going to tell you a very, very tiny snippet of the Parkinson work, because I think it so beautifully illustrates in the adult space, in the neurodegenerative space, how environments and genetics can potentially interact, and more importantly, how potential interventions can make a difference. And this work actually started as one of those nice stories that I'm sure many of you have been part of. When my very first PhD student, who returned to Germany and became a group leader and got a grant to look at Parkinson's disease and environmental enrichment. And so we collaborated, and Sam Schaffner and my lab has now taken this on, really sort of put this idea of the epigenetic view on Parkinson's disease in context. And again, as I said, it's well known that in Parkinson there is a genetic component to it. There's the very rare, sort of very strong variants like LARC2 and things like that, and alpha-synuclein, and many sort of more single nucleotide polymorphism, polygenic effects. But there's also a clear role for the environment. So exposure to things like pesticides and all that have a detrimental effect. Smoking, presumably, is good for you, as is exercising and other things. And collectively, that then affects the onset and the severity of Parkinson's disease. And Sam has sort of postulated that to some extent that converges on these DNA methylation, hydroxymethylation of these dimmer switches. And key to understanding Parkinson's is a protein called alpha-synuclein. It has many roles, but one of them that actually does interact with many of these sort of players that make up that epigenome that I showed you earlier. So, for example, various enzymes that put on various groups onto its histones, but also on DNA methyltransferase itself. And so what Sam started out doing is to use a human cell line that overexpresses alpha-synuclein to just even test, does that actually affect DNA methylation? And this cell line is a neuronal cell line that has been, a fetal cell line has been differentiated into neurons. And long and behold, in work that we published last year, together with Diego Otero at the University of Göttingen, Sam could show that there's a very substantial effect on these LUMI cells that are differentiated into dopaminergic neurons that is dependent on overexpressing this alpha-synuclein. So then, having sort of been encouraged by this, Sam went back and said, can we, in many of our human population studies, it's really hard to look at mechanisms and to look at cause and causality versus correlation. And so Sam, together with Julia Schulze-Henrich, my former PhD student, recently embarked on a mouse study. And Dr. Leong will probably recognize this mouse. So this is this idea that and in environmental enrichment, the idea that in these alpha-synuclein mice, generally they're housed, like you can see on the left here, in this sort of atrocious little plastic boxes. But there is a paradigm for environmental enrichment that you can see on the right here, where these mice actually get to play, they get to hang out with each other, they get to exercise. And to some extent, that sort of recapitulates what we know from humans, and that, you know, this sort of engagement, cognitive engagement, physical engagement actually helps the sort of buffer up the effect of Parkinson, or maybe delay its onset. And so these mice recapitulate this. And then in a very complex study, Sam was basically asking, can I basically understand why this environmental enrichment in these transgenic mice has these effects? How do they get under the skin? And I don't have time to go on all of this, but this paper is currently under review. We're very excited about it because here we went beyond DNA methylation. We looked at many other of these epigenetic marks. We also looked at gene expression, and we compared basically mice that are transgenic to wild-type mice and or not having the enriched environment. And so to make a long story short, what we find here is that's actually the genetics that's more powerful driver of the methylome than the environment itself. And so I'll leave that here. We can happy to talk about it later. But I want to switch gears slightly and just basically everything I've shown you so far is, of course, an epigenetic association. What it means is that, we identify certain pathways, certain dimmer switches, certain pathways like the inflammatory pathway. One of the problems is that the epigenome basically is highly tissue specific, because from the moment we're conceived, it is actually involved in differentiating tissues. It is involved in maintaining tissue differences. And so that makes it a bit difficult for those of us, for example, who are interested in the brain, like in Parkinson's disease, because, I would venture to say that very few of you would donate a piece of their brain to me. If you want, please see me afterwards. And so we're really sort of stuck with tissues that we can easily ascertain, like blood and cheek swabs and such. It's becoming statistically more and more difficult because we're dealing, with more and more of these CPGs and these dimmer switches. When we started this in 2007, we measured 1,536. Now we measure 1 million. You can measure all 28 million if you have your fancy machinery, and so that creates all kinds of statistical problems of false positives and such. And so because the epigenome changes over age, folks, in particular Steve Horvath, who used to be at UCLA, now is at Altos Labs, came up with the idea, can we harness this? Can we exploit the fact that these marks change during our age to basically come up with a clock that measures the epigenome? And so Steve did this, and without going into too much detail, that the granddaddy of this clock is called a Horvath clock, which came out in 2013, so about a decade ago. And he used machine learning to basically create a predictor of epigenetic age. And the simplicity of it, he actually trained it on all different tissues, so it's not so tissue specific. And the simplicity statistically, it's you have one number, and you basically just need one number. So, well, even I could potentially do it in Excel, the analysis, potentially. And so how does this look? In the DNA method, in the Canadian longitudinal study of aging, my former staff members, David Lynn and Lisa McEwen, they looked first at the association. So this is an association that I've shown you before with age, and you can see again many of these dimmer switches changing with age in relation to age. So that's the epigenetic association. We take it very same data set and we apply these epigenetic clocks. And what do we see? Well, we see these are two of the sort of more historical clocks, the Horvath clock, the granddaddy, and the Hahnemann clock. And you can see that there is a correlation between chronological age and the computed epigenetic age on the y-axis. You can also see that there is noise in there. And some of this is actually real data as opposed to technical noise. And what we then can, of course, ask, what's the difference between the chronological age and your methylation age? One of the things that we repeatedly see, and this is good news for the females in the room, is that females tend to be epigenetically younger than males, as you can see on the bottom here. Now, again, Meg Jones, my former postdoc, put this in the context, and it's like, how do we can think about this? So if we look at the vast majority of people, or not the vast, the majority of people, that would be probably in this concordance zone where your chronological age and your epigenetic age are somewhat correlated. So you would be in this concordance zone. There might be some folks whose epigenetic age is much older than their chronological age, or some folks like Dr. Leong, who I would anticipate his epigenetic age to be younger than his chronological age. And so when we then look at this in the context of health, of course, the folks with the epigenetically young phenotype would be much more healthy than those with the epigenetically old clocks. This has been shown now in many, many, many different conditions, too many to mention. We have looked at it going back to our original hypothesis in the context of early life socioeconomic disadvantage, together with Greg Miller and Edith Chen, showing that folks, again, who grew up in low socioeconomic status have an accelerated epigenetic age, so they're epigenetically older than their chronological age. These are people now in their 40s or 50s. And in contrast, folks who grew up in high socioeconomic status have an epigenetically lower age compared to their chronological age. And again, irrespective of their current age. We have also sort of ventured into the blue zones of the world. I don't know how many of you know the blue zones. These are 4 areas where people don't just live very long lives, they live extremely healthy lives. So the health span is actually really amazingly well. There's people in their hundreds that still make their family meals, that still tend their olive vine or their vineyard, the garden part of their community. You can see the poor here. We've been primarily focusing on the one in Nicoya in Costa Rica, together with my good friend David Rieckhoff at Stanford. And again, asking what can we see under the skin? Why is it that these people in these blue zones live such healthy life compared to even age-matched people that are outside of the blue zones? To our surprise, in a paper that my brilliant former PhD student Lisa McEwen published, there was actually no epigenetic age differences. So the clocks weren't actually differences between the people who lived in the blue zone versus age-matched people outside of the blue zone. However, what we found is that people in Costa Rica tended to be 7 years younger epigenetically than their chronological age. This we replicated several times now. So there's something in Costa Rica that keeps folks epigenetically young. Or alternatively, there was just no Costa Ricans in the training set, and it's an artifact of machine learning. But what we did find is that because we can actually predict from these methylation profiles, not just the epigenetic age, we can also predict immune function and such. And what Lisa found is that these Nicoyans actually tend to have a higher portion of young immune cells. So naive cells that hadn't been exposed to a sort of a pathogen. And so the idea is that actually perhaps why these folks age so well is that they haven't been so sick as much, or they haven't been exposed to much of the bugs. And so really fascinating work. More recently, we've ventured into the Italian and Greek blue zones. Oops, I skipped this, where again, this was a bit of a challenging study because they forgot to collect control groups. So my wonderful PhD student, Hannah Ruth Engelbrecht, she made, as they say, lemonade out of lemons and compared females and males in these blue zones in Nicaria and Sardinia, and finding, again, epigenetic age differences between men being epigenetically older. There's much more to it, but I wanted to highlight this study because it's actually the very first study that we published under the affiliation of the Edwin S.H. Leong Healthy Aging Program at the time, now, of course, the Center for Healthy Aging. And we're excited about this study and continuing this line of research. What Hannah Ruth found is that there are a number of things that we can also compute, not just, again, immune function. We can now also compute things like inflammation indexes, smoking scores, telomere length. And again, we're seeing differences in some of these measures between men and women, but not in all of them. And we're currently trying to sort out how they relate to health outcomes. Now, one of the things we noticed a long time ago that these clocks work very well in adults. They work very poorly in children. And so Lisa, again, teamed up with Steve Horvath. And some years ago, we basically took all our data that at the time were available and put them together into a machine learning exercise to create a clock that would work very well in children. In this case, we focus on buccal cells because that's what we mostly can ascertain for children. And what you can see here is that we train the clock to work very well, and in the test data set, it works very well as well. And so we're now sort of trying to put this clock in the context, looking at genetics and underpinning in sex, gender, physical and social environments, and health and developmental outcomes and how they relate to the clock. And importantly, we're also focusing on interventions. So if anyone of you is interested, please be in touch and become a partner in this journey. We're really excited about this. Just going to give you a small flavor of what we already have done, that work that actually is very closely linked to sick kids here. So in work with Stephen Miller and a small postdoc Noah Goma, who's now at Western, we recently shown that this pediatric epigenetic age does associate with adverse neonatal brain growth and also neurodevelopmental outcomes in children that were born very preterm. As you know, Stephen is sort of an expert in these very preterm babies, and really fascinating that, especially if we look at their term equivalent, we see these association that these children tend to be epigenetically older, that were born very, very preterm compared to extremely preterm compared to very preterm. Of course, it's yet another study where it's hard to ascertain controls, but we're now continuing to collaborate with Stephen. As you know, he's now in Toronto, and excited especially about the things, not just with brain development, but also cognition and language, the associations in this early study. In unpublished work, we also find an association with obsessive compulsive disorder, which tends to be associated with accelerated epigenetic age in these children. Now, I should say that we still don't know, and it's early days, whether we know in adults it's actually generally not good to be epigenetically older. In children, we actually don't know. I'm curious to talk to the more developmentally oriented folks in the audience, whether you feel it's a good or bad thing to be epigenetically older in children. And so, I think hopefully I've given you just a tidbit of our work that really points to the fact that experience and environments do associate with epigenetic patterns to affect phenotypes in health, using a paradigm, a slew of all kinds of different environments, all the way to these blue zones from prenatal stuff, that we need to look at this in the context of development, and that we need to look at this, which I didn't really have a chance to talk about too much, but as I briefly mentioned with the Parkinson, in the context of the genome. And so how does this all then underlie our concept for the newly established Edwin S.H. Leong Centre for Healthy Aging? And so Dr. Leong has set out the challenge for UBC some years ago, I think 3 years ago, Dr. Leong, 2 years, 3 years? More than that. Really, and then you can see here the sort of things that were, Dr. Leong's vision. And you can probably, when you read through this, you can probably see just based on my talk that really spoke to me tremendously, and especially these things like life course and transcending disease mortalities. And so when we started out a year ago to build this center, and at UBC it takes a rigorous 9 meetings with all approvals and many different things to become a center, and we just finished it. So thanks to my wonderful research director, Kim Schmidt, who is with us. We basically then decided that our center will focus on 4 core themes, that being gerosciences, so the biology of aging, with the idea being, as Dr. Leong proposed, we want to transcend disease mortalities. It's clear, the concept of gerosciences, that the process of aging is the highest risk factor for age-related diseases. And so understanding that biology is really key. As you can tell, I love the things going from what we see in society, drilling all the way to the cell. We, of course, want to prevent and intervene so that people live a healthy and long life. And we want to also, of course, collaborate with our friends who are very focused on age-associated diseases, but really bringing that lens of geroscience society cell to that conversation. All of this, of course, we want to wrap around with impact and implementation. We realize there are great opportunities for education, teaching, and learning, and we are deeply committed to respect, equity, diversion, and inclusion. And not just in sort of the way the people we hire, but also really in our research. And I think there's great connections to the center here, the Edwin S.H. Leong Centre for Healthy Children, because one of the key areas that you could probably guess from my talk are really social determinants of health. In our case, the social determinants of healthy aging, and I know you folks are heavily focused on inequities and such. So I think there's a nice story arc that we can develop. Cross-cutting to this is a deep commitment to interdisciplinary research, collaborative research, and the life course. And so we already have started to build our center very quickly in that sort of interdisciplinary framework. You can see here we have members from 8 different faculties across the 2 campuses of UBC. UBC, for those of you who don't know, the main campus is in Vancouver, but we have an outpost in Kelowna and Okanagan, ranging, of course, rooted in the Faculty of Medicine, but not restricted to the Faculty of Medicine. We have the Faculty of Arts, Faculty of Science, Education, Applied Sciences, Pharmaceutical Sciences, and even Dentistry in our group, as well as Health and Social Development at the Okanagan. So very proud of the enthusiasm, the excitement that was created by the announcement of this initial program in our center, and the buy-in we've gotten to really build an interdisciplinary program. And this idea of the life course is really rooted in the fact that we think that aging is a lifelong journey. I've told you a lot in today's talk about this biological embedding of things like socioeconomic status, stress, and so on, that gets into our DNA, gets into our cells, and determines to some extent our current health. But of course, we can then also look forward and say, who are we now today? How can we predict how we age? And we're focusing here on things like multi-omics integration, like we did with Parkinson, wearables, big data, database linkages, and AI and ML, and with really a deep commitment to exploring the entire life journey and the life course. And in part, this commitment is really also to understand when do interventions that you see here at the bottom, when are they best, when do they make the biggest difference, when do they have the highest impact. And so we're already working on a number of different interventions, physical interventions, psychosocial. We're actually just got an NIH grant to look at economical interventions in people of low socioeconomic status and how they affect health across the lifespan. And again, one of the major tools in our toolbox are these aging clocks, not just epigenetic clocks. We're looking at many other clocks, brain clocks, microbiome clocks, and so on. And the focus of our center is, as I mentioned before, on health span and not just lifespan. And we're, of course, trying to find that delicate balance between population health on the one hand and individual health and potentially precision medicine on the other hand. And so with that, we have come up with our first initial research strategy priorities, that being faculty growth. We already have lots of faculty, but we've got some new positions already committed by UBC that we're currently hiring for. So if anyone knows young, ambitious postdocs who want to get into the aging space, please be in touch if at least one or maybe two positions. We're, of course, working, despite that very generous gift from Dr. Leong that's just launching the rocket. We need to work on funding and sustainability. We have a deep commitment to knowledge exchange. We have public series, lecture series for the lay public. I think a great opportunity for us to interact between the two centers, really training and education. We already talked to Stephen Miller about this idea of, having our trainees, the ones that are focused on early life, get an appetite for later life, and the folks who are focused on later life get an appetite for earlier life. And so looking forward to some hopefully rich conversations. And we're also deeply committed to developing partnerships, not just nationally, but also internationally. So we already have signed partnerships agreement with Stanford, Columbia, and the Pasteur Institute to really collectively bring together experts in the field of healthy aging. And so with that, I hope it's a bit of a crazy journey, but I hope I give you a bit of an idea where we're at. And I encourage you to participate, to work with us, and please be in touch with either Kim or myself. And so just want to acknowledge, of course, all the trainees and the wonderful staff that over the years have allowed us to do this work, and the great collaborators, the funding agencies, and last but not least, the wonderful generosity of Dr. Edwin Leong and the Tai Hang Fei Charitable Foundation to allow us to launch that rocket of the Edwin S.H. Leong Center for Healthy Aging. Thank you very much. Oh, it's almost in time. Perfect.
Appreciate the wonderful lecture, Dr. Kobor, and really highlighting that aging begins in early life, and really the focus on the integration of a life course approach, which really I think speaks to many of us in the room. I'd like to open up the floor for some questions. Dr. Buda.
Brilliant talk, Mike, as usual. So thank you. There's a lot published now on adverse childhood experiences earlier on. And one of the big conundra is really around the additive nature or the multiplicity of those experiences. Is there any epigenetic imprint that allows us to understand if poverty alone, or if poverty is mixed with other adversities like conflict and other things?
Yeah, that's a great question. And I'm going to take a slight detour, Sophie, if you permit. One of the beauties of working in an interdisciplinary space is that everyone approaches the problem differently. And so what we have done, because we're sort of coming from the data science, we like the data to speak to us. So we have things done like, principal component analyses of various environments, and we identify things that potentially by our correlations hang together. But then my friends from psychology say, oh, no, you can do this. We know from literature from 1970 that they don't hang together. And so we're sort of caught in a little bit in that sort of no man's land between, what do the data say us without sort of stepping onto the toes of the expert. That said, of course, a lot of these things do hang together. And I think what we're hoping to do is to identify populations where we can isolate some of these effects. So I'll give you one example. We have a collaboration with the University of Utah on sort of prenatal exposures, and actually it turns out there's a lot of prenatal drug use, but no alcohol. So this is where we can take alcohol out of the equation, for example. And there's many of those, but it is not easy. On the flip side, of course, you could think about, can we model some of them, these adverse, in animals? Very, very hard to do, right? Especially when you talk about poverty, when you talk about some of this more human-specific things. So it's early days. I think what we ultimately like to come up with, and that's where we make some progress, is what we call the methylation risk scores that affect, that are specific for particular exposures. And then putting this all together in a larger data frame to see. I suspect, there will be some of these associations that are true across the board versus others that will be specific for a given exposure. A great question, yeah.
That was an amazing talk. I wanted to follow up a little on Zulfi's question and your answer. If you look at specific populations and you hypothesize that these interventions will make an impact on the outcome, is there data yet to show that some of the dimmer, the epigenetics changes with them? I mean, it's obviously hard to do in these large scale studies, but if you look at a particular disease or outcome, can you actually see the white cells changing, the signaling cascade for NF-kappa B changing, along with positive outcome to show that it's a biomarker?
This is a great question. And so the dirty secret of a lot of the stuff I'd shown you are that these are not studies that were designed to do epigenetics. They were studies that were designed to do genetics. And so I say the vast majority of what I showed you were piggybacking on people who had collected DNA once. Of course, to answer your question, you would want to have DNA before and after a set intervention. Now we're having some, or at least in a sort of, maybe even if it's just after, but it's a randomized trial or something like that. So we're getting there. The child cohort that many of you know here is ideally we have, a few measures. We are doing, we have a collaborator in Bangladesh who's a randomized control trial on early life nutrition. We have published a small study on elderly folks who in a nursing home that had a sort of a walking intervention. And so there's but for this to be most rigorously done, we need, and so we're getting away from the piggybacking and, maybe 5 years ago started to design studies where we have multiple measures. So stay tuned next time I come to this.
Okay, thanks.
Somebody there.
Can you just speak up?
Yeah, it's a little tricky again because it comes back to the same question that you want repeated sampling. I tell you one thing, and I don't know whether Ryan is here. Ryan.
Atrium zone 694.
For those not from here, that's the main hospital, not here.
Yeah.
Go ahead.
Oh, okay. So Ryan, was on this committee when he was in UBC, and then he went to Steve's lab as a postdoc. He actually sort of determined already the methylation patterns in fetuses, and it actually turns out that they're already quite well established. And so, it's not that you're born with a blank slate because of the tissue difference. And so the question is, and it goes a little bit in the same direction to Sophie's question, how much space is there for these environments? And, again, there is plenty of evidence from child development that there are these sensitive periods early in life, but I think it's a little bit like looking under the lamppost. I think, we all sort of remember probably when we went through puberty and adolescence as massive changes. And so that could be another window where, we're working now with Megan Gamma and Ian Gottlieb at Minnesota and Stanford respectively to look before and after puberty. Are there, methylations or that people talk about an age plasticity window. So I think, the world is your oyster if you want to sort of embark on this, with the challenge that you need samples, of course, ideally for many, many different years. But good question, yeah.
So on behalf of the Edwin S.H. Leong Centre for Healthy Children and the Research Institute at the Hospital for Sick Children, thank you again, Dr. Kobor.
Thanks for having us.